Hi guys, today we're going to be talking about microbial weathering again. And in this part 2 video to microbial weathering, we'll be covering sulfide oxidation, bioleaching, and biomining specifically. Last video in the part 1 microbial weathering video, we went over mineral dissolution, mineral surface reactivity, microbial colonization and organic reactions, silicate weathering, carbonate weathering, soil formation, and weathering's impact on global climate. And in this video, we were going to cover both sulfide oxidation topics as well as microbial corrosion, but since we have a lot to cover in those topics, I'm splitting it up now into 2 videos. So there will be a part 3 video after this one over microbial corrosion. But in this video, we're covering sulfide oxidation, specifically pyrite oxidation mechanisms, biology's role in pyrite oxidation mechanisms, since this is microbial weathering, and bioleaching and how we can employ bioleaching to mine for metals. So the reason that we're focusing on pyrite oxidation is because pyrite is a really common sulfide mineral. And when pyrite is exposed to oxygenated waters, both iron and sulfide can become oxidized because currently in their pyrite form, they are reduced, just meaning that they have a lot of electrons and they can give some away to become oxidized. And that's what oxygen does. It takes those extra electrons and it oxidizes the reduced iron and sulfur that are within the pyrite. And when it oxidizes pyrite, pyrite dissolves. And this behavior of pyrite, as well as its distribution and chemical transformations in the environment, are really important for us to understand because pyrite is an extremely common constituent of coal seams, ore bodies, and shales, as are other sulfide minerals. And so we'll talk later about the importance of mining sulfide ores and how we can even use biology, microbes, to help us in this process. So let's talk a little bit about how pyrite oxidation occurs, what mechanisms are involved, and kind of walk through this step by step. So pyrite goes through a series of redox reactions when it's becoming oxidized and ultimately dissolved. First, we can see here that oxygen does attack these sulfide ligands, the reduced sulfur ligands at the pyrite surface, that it can oxidize. And the second step to oxygen attachment is the breaking of the oxygen-oxygen double bond and the displacement and removal of the sulfur hydroxide that forms due to this oxygen and water attack. And then these sulfoxy anions are released into the solution. And once in solution, they are oxidized further to sulfur intermediates such as thiosulfate, tetrathionate, trithionate, sulfite, and finally the final most oxidized sulfur compound, sulfate. But the sulfur is not the only thing that's becoming oxidized. Iron also becomes oxidized because iron is also in its reduced state, iron 2, and the 2 here is just its oxidation state, which for the purposes of this video, you don't really need to understand. All you need to know is that iron 2 is reduced or ferrous iron, and iron 3 is oxidized or ferric iron. And so the iron 2 in the pyrite is oxidized by exposure to air, and then it forms iron 3. And the iron 3 can be incorporated into ferric hydroxide minerals. And so this is a solid mineral that forms when iron is oxidized to iron 3 and hydrolysis occurs. And then step 3 is basically that because iron 3 oxidized ferric iron forms, that ferric iron can then continue to oxidize the rest of the reduced iron and sulfur compounds that are in and surrounding the pyrite. However, I mentioned here that iron oxidation mechanisms are heavily dependent on pH. I meant to mention that earlier. Oops, I'm sorry, I'm mentioning it now. So because they're heavily dependent on pH of the ambient solution, when the pH is greater than 4.5 or around that, the ferric iron likes to be in the solid phase as ferric hydroxide minerals. And because it's in the solid phase, it's less efficient of an oxidant to pyrite than it would be as dissolved iron 3 ions. And so remember that when pH is lower than 4.5, dissolved iron 3 ions are really, really efficient oxidants of pyrite. And so they just go and dissolve the pyrite really quickly. Whereas ferric hydroxides, which adsorb to the surface of pyrite, can act as oxidants and dissolve the pyrite directly, but it just isn't quite as efficient as if the iron was fully dissolved, and that only happens when the pH is super low. But there are two main factors on this slide that make these pyrite oxidation and dissolution mechanisms a self-sustaining cycle. And these two things are, one, that the oxidation, dissolution, and removal of iron from the pyrite surface exposes more reduced sulfur, and this causes then the sulfur to become oxidized again, like we showed on the previous slides, exposing more iron, which then becomes oxidized, and so on. And then two, when the pH drops below 4.5, iron 3 dissolves and becomes the most efficient oxidizer of pyrite, even more efficient than oxygen. And you might be wondering, what if the pH is above 4.5? How is the iron going to dissolve and become more efficient at pyrite oxidation at that point? Well, this is where the magic happens. Because both sulfur oxidation and ferric hydroxide formation leads to the release of hydrogen ions, this decreases pH, because we know that pH is just a concentration of hydrogen ions in solution. And because this decreases pH, the pH drops below 4.5, and this promotes iron 3 dissolution and more pyrite oxidation. And this is just a self-promoting cycle that continues to snowball down this ramp until the pH is extremely low and all the pyrite has dissolved. So what, if any, are processes that might hinder this process, since it seems like everything's causing this snowball effect? Well, there are a couple things that the increased acidity causes that hinders the continual oxidation and dissolution of the pyrite, and that is decreasing pH increases the stability of elemental sulfur, causing precipitation of this elemental sulfur crust around the pyrite, as shown down here. It's kind of like a protective layer that doesn't allow any further ions to go and try and oxidize what's below that elemental sulfur surface. However, if there are sulfur oxidizing bacteria present, and now we're getting into the microbial oxidation processes and microbial weathering, like the topic of this video, if these types of bacteria are present, they could eat away at this sulfur crust layer and allow further dissolution of the pyrite. Moreover, in acidic conditions, pyrite can reduce iron 3, so ferric iron can not only reduce pyrite and dissolve it, but pyrite can also act to reduce ferric iron. And so they're kind of this trade-off mechanism where, okay, I give you an electron, but then someone has to take it, right? And so there's not only things getting oxidized, but also reduced in this process, and that's why they're called redox reactions. And because of this, pyrite's also reducing the ferric iron, and this can hinder the process of ferric iron oxidizing and dissolving pyrite. Therefore, if iron 2 oxidizing bacteria are present, they can continually produce more ferric iron with any ferrous or reduced iron 2 that's formed, and that's because they use the redox reaction of ferrous iron oxidation to ferric iron as their energy producing metabolic pathway. And so they're producing all this iron 3, the sulfur oxidizers are oxidizing the elemental sulfur crust that's preventing further pyrite dissolution, and both these types of bacteria can really help to catalyze the pyrite oxidation and dissolution process. So we just mentioned a few ways that biology can play a role in the oxidation and dissolution of pyrite as well as other sulfide minerals. But what exactly are they doing? Well, they enhance oxidation rates. So we kind of just mentioned this. They enhance oxidation rates because iron 2 oxidizing bacteria are not only acidophilic, meaning they love to live in acid environments, but they also generate ferric iron much faster than any abiotic iron oxidation process can. And so they generate this continual flow of ferric iron to that pyrite surface for its oxidation. Another byproduct of this ferric iron production is that this ferric iron is formed in acidic conditions, obviously because this snowball effect causing the acidic conditions is just getting more and more acidic as iron and sulfur are oxidized. oxidized. And because of this, the ferric iron is soluble and reactive and can effectively oxidize reduced sulfur as well as iron at the pyrite surface. And when iron 3 acts on something else to oxidize it, like reduced sulfur compounds, it then becomes reduced back to iron 2 or ferrous iron. And because this is what's feeding those acidophilic bacteria that are producing the iron 3, this keeps those guys alive and it keeps the cycle going. So again, it's a self-sustaining cycle. It's really, really cool. But because iron oxidation as a metabolic pathway for these bacteria does not produce that much energy, these bacteria must oxidize large amounts of iron all the time. And so because of this, it's unsurprising that measurements show that these bacteria account for the majority of pyrite dissolution in many acid mine environments. But continuing on talking about biology's role in pyrite and other sulfide minerals oxidation, not all acidophilic bacteria that are present in these types of environment oxidize iron too. Some also oxidize reduced sulfur. We talked about these guys being able to eat away at that elemental sulfur layer and continually expose more underlying pyrite surface for further oxidation and dissolution. And so these guys are called sulfur oxidizing bacteria. But some of these bacteria do both iron and sulfur oxidizing. So these guys living in this environment can really speed up the process of pyrite oxidation and dissolution. And this bacterial sulfur cycling ultimately leads to increased acidity, further continuing this cycle of increased acidity, increased bacterial activity, increased pyrite oxidation and dissolution, etc. So again, it's this snowball cycle. There are a couple things that kind of slow it down because again, whatever's becoming oxidized also reduces something, etc. So there's other things that are going back and forth, but in general, it's this snowball effect causing more and more pyrite dissolution till it's all gone. But it's important to remember that there are two major ways that these bacteria, these iron and sulfur oxidizing bacteria, can contribute to pyrite and other sulfide mineral oxidation and dissolution. And these two ways are indirect and direct. The indirect bacterial pyrite oxidation pathway occurs because the byproduct of the bacteria's activity, like producing iron 3 because you oxidize iron 2, is then abiotically oxidizing the pyrite. But there are also bacteria that directly oxidize and dissolve sulfide minerals like pyrite, and these are iron and sulfide oxidizing bacteria that grow attached to pyrite and sulfide mineral surfaces. And this attachment allows them to directly oxidize and dissolve the reduced iron and sulfur compounds via enzymatic reactions. But the mechanisms behind this direct attachment of these directly oxidizing sulfide mineral bacteria is important, and it happens in 4 steps. This attachment happens by the transport of the bacteria to the mineral surface, the electrochemical interaction of the bacteria with the surface, the attachment of the bacteria onto the surface by fibrils and or EPS, which stands for extracellular polymeric substances, and the multiplication and eventually the development of a colony at the surface. And so these are the 4 steps that the bacterial attachment involves. And the 3rd step we can see here, there's a couple nuances. So once attached, the EPS or extracellular polymeric substances, basically like sticky enzymatic mucousy substances that the bacteria excrete, these EPS substances fix the cells to the surface of the mineral. And because they absorb iron cations, because it's negatively charged EPS, they can act as electron shuttles, these iron cations in the EPS, shuttling electrons from the mineral to the cell. But in terms of these 2 major types of sulfide mineral oxidation mechanisms, it's still uncertain whether direct or indirect pathways are responsible for the majority of sulfide mineral oxidation. But in general, it's thought that the more reactive sulfide minerals, such as pyrrhotite, are oxidized predominantly by indirect mechanisms. So just iron oxidizing bacteria produce ferric iron, which then abiotically oxidizes and dissolves those sulfide minerals. However, the less reactive sulfide minerals like pyrite, which is really stable and not very reactive, are oxidized mostly by direct mechanisms. But now let's switch gears a little and talk a little bit about why this is all important and how maybe this matters in an environmental sense. So in mining, waste and tailings are often discarded in soils and ponds. And because they contain pyrite and other sulfides, they then become oxidized by natural processes like we've just been talking about, and this creates a highly acidic, sulfate-rich, metal-rich effluent called acid mine drainage, or AMD. And acid mine drainage is a major environmental concern. And I have here listed 4 major reasons why it is environmentally very concerning. And this is because it's acid mine drainage, so it's acidic, and that means it's corrosive. It is very metal rich, and some of these metals are really toxic to aquatic life, as well as the acid can be toxic to the aquatic life. And 3, because ferric hydroxides and ferric sulfates form due to the oxidation of the iron by iron oxidizing bacteria, these minerals begin to coat benthic species and inhibit photosynthesis. And lastly, this acid mine drainage often converts forms of dissolved inorganic carbon, like bicarbonate, into carbonic acid, making it less bioavailable for, again, aquatic life. So all of these things are environmentally concerning, especially for aquatic life, which can be severely impacted by acid mine drainage. And so this kind of leads us into the discussion over bioleaching and biomining, which you might think, okay, biomining, we're mining for metals using biology, employing biology's ability to take up metals, which it does have this incredible ability. And I've talked a lot about this in my bioremediation and biorecovery video. But it not only is used for targeting and collecting valued metals, but also for taking up potentially toxic metals, just like we saw in the acid mine drainage. So environmental remediation as well as metal recovery can be achieved by something called bioleaching. So now moving into this topic, I say here, what is bioleaching? Well, I kind of just explained it. We're using biology to target and take up metals. However, in bioleaching specifically, metals are dissolved and the solution is collected and then reprecipitated at a plant, and the solid residue is discarded. So that's kind of how bioleaching works. And why do we need to bioleach, or why is it economically viable option for mining and remediation purposes? Well, in many sulfide ores, there are very valuable metals, which we'll talk about on the next slide. And because sometimes they're present at pretty low concentrations, it makes traditional abiotic extraction mechanisms really inefficient and economically just not financially smart. So because of this, we employ bacterial oxidation. So just as we talked about in this whole first half of this video, we talked about how bacteria can oxidize pyrite, meaning they oxidize the iron and sulfide, and then the iron goes into solution. Just like this, we can also use bacteria to oxidize and put into solution metals such as copper, zinc, lead, arsenic, antimony, nickel, and molybdenum, all from sulfide ores. And I'll talk today about 2 common methods of bioleaching. These include dump bioleaching and heap bioleaching. Dump bioleaching is when we add acid to waste rock and indigenous bacteria oxidize the sulfide mineral residues. And this is kind of a slow and inefficient process. Because of this, we also have the heap bioleaching method. And this heap bioleaching is done by placing finely crushed ores onto prepared pads and spraying sulfuric acid onto the heap of finely crushed ores you just placed on the pad. And this is to precondition for the bacteria. So again, we're talking about acidophiles here. They love acidic environments. And because of this, we can use sulfuric acid to precondition for the bacteria. And then the liquid that comes out of the bottom of the heap from the prepared pad that we talked about is then collected, and this collected liquid is transported to a plant where the metal is reprecipitated and purified. But one of the main things that affects the efficiency of bioleaching is the maintenance of bacterial growth, because again, we're just kind of letting those indigenous bacteria that like the acidic environment and want to oxidize and dissolve those metals take hold and do their thing. But what contributes to maintaining their bacterial growth? Because I can tell you, I culture bacteria in the lab, and it takes a lot of work just to keep them, living. Well, mine are actually pretty robust, but still, living to the extent to where they're being efficient takes a lot of work and maintenance to keep them at that high growth rate, because you want to keep the rate really high so that you're still gaining this economic advantage of using bioleaching as a biomining technique. And so because of this, we can maintain the bacterial growth at its most efficient level by using oxidants. For example, oxidized iron is necessary to oxidize metals like copper, and because the addition of us adding ferric iron to the heap is uneconomic, we can rely on ferrous iron oxidizing bacteria to produce that ferric iron for us. That'll then oxidize the copper, and we can get the copper into the solution and get copper out of the process as well. But to maintain these types of bacteria, we must add oxygen to the pile. And so we do this by aeration processes where we add the oxygen into the pile. And for this, the pile also has to be permeable. I know this is the third thing. I'll get back to acidity, but permeability is also really important to maintain in the heap so that we can have the diffusion of air through the heap as well as solution channeling. And now back to acidity here. This is the second thing on this list. We also need to maintain the acidity of the heap to keep the bacteria growing at an optimal rate because they are acidophiles. The next thing here is nutrients. Bioleaching bacteria, like any bacteria, require nutrients such as ammonium, phosphate, potassium, etc. Because of this, we can add those types of compounds into the heap as well. And lastly, heat. These bacteria grow best at a certain temperature range, around 20 to 35 degrees Celsius. And because of this, we need to slightly heat up that heap to keep them growing at their best rate, living their best lives. So that is how we use bacteria for bioleaching. But because some minerals contain no redox active sources for energy, simple dissolution is the only thing needed to dissolve those minerals instead of oxidation processes. And because of this, sometimes bacteria aren't even used. Sometimes fungi are the best option. And so in this slide, we can see here that fungal acid production is actually a really effective method for the dissolution of minerals that don't really have any redox active sources and can't really be oxidized to be dissolved in the same way that we talked about previously for pyrite. And a couple other advantages to using fungi are that they can grow at really high metal exposures and they can tolerate higher pH levels than acidophiles can. And I don't have mentioned here for fungi, but in the first microbial weathering video, as well as in the next video over microbial corrosion, we did talk, and we will talk more about how fungi also have the ability to physically weather rocks and kind of crack and fracture them with what's called hyphae. And because of this, they can then insert their organic acids even further into the rock and really just break it up and dissolve it from inside out. So they're really, really efficient at weathering minerals and rocks. The last thing I want to discuss is, can we bioleach gold? And this is because I know we're probably all thinking of mining and obtaining economically valuable minerals. And because of this, we automatically think of metals like gold because these are really economically valuable and a little bit more fun to think about than things like iron and copper. And so can we bioleach gold? Well, many ore bodies do contain refractory metals like gold, but these metals are really hard to extract, which is why they're called refractory metals. And that's because they're disseminated evenly throughout the host sulfide minerals in the case of sulfide ores. And because of this difficulty of extraction of these types of metals, one promising method that has been shown to be able to extract these metals is bio-oxidation. So this sounds exactly like bioleaching. We talked about how we're using biology to oxidize and dissolve metals. So how is this different? Well, it's similar to bioleaching in that, yes, bacteria similar to what we just talked about are used to degrade sulfide minerals, but unlike bioleaching, bio-oxidation leaves behind certain metals as solids. And typically, depending on what kind of bacteria we choose for this process and what kind of composition the entire ore deposit is, we can kind of choose which metals of value we want to target, which is really awesome. So that is all I have for the topic of sulfide oxidation, bioleaching, and biomining in the microbial weathering series of videos we're having here. But in the next video, like I did mention, we are going to talk about microbial corrosion, and then in the video video following that, we're going to talk about microbial zonation, which I don't have a thumbnail here for yet, but I hopefully will soon. So stick around for the microbial corrosion video. And in this video, like always, I mentioned my reference, or my major reference at least, which is the book Introduction to Geomicrobiology by Kurt Konhauser. And this is a great book. It's linked in my description if you want to check it out, as well as other supporting references down there as well. So go check those out. And lastly, if you guys have any topic suggestions for me for the future geobiology videos or any other playlist or topics on my channel, please comment them down below. I'd be happy to take a look at them and maybe do a video on them in the future. So thanks again for watching, and I'll see you guys next time. Bye.